Okay, I think it's time for a start. So my name is Matt Tinning. I'm the head of the laboratory operations team at AGRF, and it's my pleasure to welcome you to our fourth presentation of our webinar series on complex diseases. It's also my pleasure to introduce our presenter today, Associate Professor Michael Hildebrand from the University of Melbourne and the Melbourne Brain Centre. Michael is the head of the Translational Neurogenetics Laboratory in the Epilepsy Research Centre at the Melbourne Brain Centre. Michael's research is supported by an NHMRC Centre for Research Excellence in Speech and Language Neurobiology and NHMRC project grants on the detection of somatic mutations in sporadic epilepsy and improving diagnosis, prognosis and clinical care in childhood speech disorders. The focus of Michael's research is to understand the basic neurobiology of human epilepsy and speech disorders and translate this knowledge into improved treatments for patients. The titles of Michael's talk today is Somatic Mutation, the Hidden Genetics of Epilepsy, which is based on his research into detection of mosaic variants in focal epilepsies, the most common group of epilepsies, as a step towards precision treatments for focal epilepsies. Please join me in welcoming Michael.
Thanks very much, Matt, for the kind introduction. I'll just share my screen and hopefully that will work. You can see that. So I want to thank Matt, Glenn, Claire and Desley for the opportunity to present my data today, which is at least partly based on data generated through AGRF, and I've had a long association with the institutes. And I wanted to share today some of our work on detecting somatic mutations, which we think of as the hidden genetics of epilepsy. So in terms of an outline for my talk, I'm going to give a brief background to the disorder, the association of the disorder with brain lesions, and also introduce somatic mutations to you all. I'm then going to talk about, give you one example of a strategy that we're using to identify somatic mutations using brain tissue, and that particular lesional epilepsy is known as hypothalamic hamartoma. And then in the third part of my talk, I'm going to introduce some alternative strategies to identifying these somatic mutations in the absence of brain tissue, and in particular focus on our recently published work on the Csf liquid biopsy for epilepsy. So by way of background, epilepsy is a relatively common condition. There's a lifetime risk of about 3 to 4%. It can be caused by acquired factors in about 30% of cases, such as stroke, trauma, for example. But we also know that there's a strong genetic contribution. I've left a question mark there in terms of the genetic contribution because we're obviously still determining it. But based on twin studies, the heritability is thought to be somewhere around 70%. For epilepsy. So we think that most of the remaining cases have some type of genetic contribution. Epilepsy can be divided into two major types in terms of seizure origin. So there can be focal seizures, which originate from one region or specific regions. They can be multifocal in the brain. And these account for about 60% of cases. And of those 60%, about between 20 and 40% are thought to be associated with a lesion. That's a mass detectable via imaging, and the remainder are non-lesional. The other major subtype are generalized seizures, and these are seizures that arise across different regions of the brain. In terms of the diagnosis, so this is always based on clinical data, clinical history. It's based on electroencephalography, and it's also often based on MRI imaging as well. Comorbidities are common for epilepsy, so things like intellectual disability or developmental delay, autism, psychosis are examples of common comorbidities of seizure disorders. And then in terms of the treatment, most commonly pharmacotherapy, so anti-epileptic drugs, although sadly around 30% of patients remain refractory to those drug treatments. And then surgery is actually normally very successful. In more than 80% of patients have a good outcome. The problem is that only a small minority, less than 5% of patients, are actually candidates for neurosurgery. So in terms of background for somatic mutations, we've known for a fairly long period of time now, going back to 2012, that somatic mutations can contribute to the development of brain malformations associated with epilepsy. We also have known for a while now that germline mutations can also contribute. And you can see in these figures here that sort of demonstrate the difference. So of course, the germline mutations are obviously inherited from a parent and are therefore present in every cell. You can contrast that with somatic, or also known as postzygotic mutations, that obviously occur only within the offspring. They're not transmitted from a parent. And depending on when they arise during early development, if they arise very early, they can in fact be present in all cells of different tissues. But if they arise later in development, they can be restricted. And some of the ones that we're most interested in when studying epilepsy, of course, are those that are restricted to the brain. And really the first study that showed that these somatic mutations can contribute to epilepsy was back in 2012. Joe Gleason's lab in the US published this original study on one very rare type of lesion known as hemimegalencephaly, where you have an entire hemisphere of the brain does not develop normally and is malformed. And what his group was able to show in about 30% of the patients that he looked at, and he had a cohort of 20, which was actually a lot for this very rare type of malformation, he was able to show that about a third of them had a mutation in the mTOR pathway, which is a pathway commonly subject to particularly recurrent somatic mutation in a whole variety of different disorders, including cancer. And in the last sort of 5 years or so, it's become evident it's also a major contributor to particularly lesional types of epilepsy. So what are the strategies that we can use to identify these mutations? Well, one, of course, if you're looking for a somatic variant, you need the tissue of interest. And we're really fortunate in epilepsy compared to almost any other neurological disease in that, you know, one of the standard treatments for a subset of patients is surgery, which means we get access to the resective tissue. And that, of course, allows us the possibility of picking up these brain-only somatic variants. Now, of course, it's also known that germline mutations can contribute. And as you'll see from the first part of my talk, we in fact are now finding examples where both germline and somatic mutations in the same gene can actually contribute to the development of these malformations. In terms of the germline mutations, obviously we can pick these up in blood using standard depth exome or genome-wide approaches, or indeed gene panels if we're targeting particular pathways such as the mTOR pathway. In terms of the somatic mutations, we can use similar strategies, except that we need much higher depth, obviously, because we know that these somatic mutations for brain malformations can be present at very low frequency, even down lower than 1% frequency. So we really need to maximize the depth if we're going to pick up all of these pathogenic somatic changes. And then, of course, given that these variants can occur at the same sort of frequency that we see sequence artifacts occurring, we need then very sensitive methods for targeted independent confirmation. And the approach that we mostly use is droplet digital PCR, but of course you could use targeted sequencing, amplicon sequencing, for example, to pick up, to confirm these low-level somatic variants. And in the case of germline variants or more high-frequency somatic variants, we can use Sanger sequencing as well. But really, what ideally, in the ideal scenario, is we have paired samples from these patients, DNA derived from the brain tissue and DNA derived from the blood. So I want to go through one particular study as an example of what we can do when we do get access to tissue. So I'm going to talk about one relatively rare type of brain malformation, although most malformations are rare, and that's hypothalamic hamartoma, which is associated with gelastic epilepsy or gelastic seizures. So these are benign tumors of the hypothalamus, and they're benign because they develop early in life and they will grow for a period of time, but then they stop growing. So they're not malignant tumors. In terms of the age of onset of symptoms, and in particular seizure onset, which is usually the first symptom that these children will exhibit, the mean onset is 10 months. So it occurs fairly early in life. The major type of seizure is gelastic seizures. These are laughing episodes that manifest as laughing episodes. They can, if untreated, go on to become generalized seizures across the brain, and then worsen further to develop into epileptic encephalopathy, where in addition to the seizures, you then have a delay in development associated with the condition. In about 30% of cases, there's also precocious puberty is a feature, and that's because depending on the location of the tumor within the hypothalamus, it can disrupt the normal endocrine functions that the hypothalamus is involved in regulating. Now, fortunately for the children with this devastating condition, back in the late 90s, Jeffrey Rosenfeld, who was a neurosurgeon here in Melbourne, developed the first approach to surgical treatment. So the hypothalamus, as you can see from the diagram here, is located in the very center of the brain. It's a difficult and dangerous region to access surgically. And so back in the late 90s, Jeffrey Rosenfeld developed an approach called the transcallosal approach, and he was able to successfully resect out the tissue. And it had remarkable success, and more than 50% of patients were actually able to achieve seizure freedom. So these patients went from having a devastating outcome in terms of epilepsy and development to having a seizure freedom in more than half of cases. The other advantage was, in addition to the obvious benefits to the patients, were for researchers like myself, we were able to get access to that resected tissue to try and understand the molecular causes of these malformations. And so using a large historical series, and I say large because the incidence of this particular tumor is very rare, about 1 in 200,000. So we had, because the surgeries were done here in Melbourne for a long time and nowhere else in the world, we had a large series of patients with tissue-derived DNA available. And we actually had enough DNA left back in before 2016 when we did this study to interrogate 38 patients with paired brain and blood-derived DNA available. And at that time, we were able to show that a number of different genes within the sonic hedgehog pathway, a very important pathway for brain development, we showed that variants in those genes could in fact cause the sporadic form of these hypothalamic hamartoma tumors. And so we've in fact implicated 17 genes. This was work published in the American Journal of Genetics in 2016, including GLE3, which is the major contributor thus far, but also other genes in the pathway such as PRK-ACA, which is one of the major upstream regulators in the pathway, as well as other downstream genes as well. In addition to the genetics, we also did some work which is actually unpublished, it's not in the paper, but we did some in vitro work to show modeling the effect of some of these variants using siRNAs in vitro. We're able to show that in fact, when you disrupt these genes, you do get an upregulation in sonic hedgehog signaling. And we believe it's this upregulation in signaling, this abnormal hyperactivation of the pathway that leads to the development of these tumors, which are malformed in the sense that they have an abnormal constitution of both neurons and glial cells as well. So from that earlier work, we were able to show that somatic variants in this sonic hedgehog pathway could contribute to sporadic hypothalamic hamartoma. So in our more recent work, we became interested in the potential contribution of cilia genes to these tumors. And that was based on a paper that came out in 2019 from a Japanese group. And they also studied some sporadic HH cases. And they in fact reported 2 with double hits, biallelic, germline, and somatic variants in this gene DINK2H1, which is a known cilia gene. And this was interesting because we were well aware that the sonic hedgehog pathway actually relies on the primary cilia for trafficking to enable its signaling during development. So biologically, it seemed like a plausible mechanism, despite the fact that it was only reported initially in 2 cases and only in 1 cilia gene. And of course, the cilia are present on almost every cell in the body, including on neurons. And within each of the cilia, what you'll find are this constellation of microtubules, and they form structures known as the IFT subunits, dynein subunits, for example, that are involved in what's known as intraflagellar transport. And they're responsible for trafficking a whole range of molecules, including sonic hedgehog pathway proteins, up and down the cilia in different cells during development. Interestingly, when you look at the Ciliopathies, these are the diseases associated with dysfunction of the cilia. There's more than 30 of them that have been described, and more than 200 genes associated with those syndromes. So it's very phenotypically and genetically heterogeneous. When you look at the human Ciliopathies, they're all, in fact, or mostly recessively inherited syndromes, meaning that, of course, both genes have to carry a variant in order to manifest the phenotyping, to manifest the phenotype. And so, and the other interesting point is that there's sort of 2 types of Ciliopathies, those associated with disruption of ciliogenesis, which is the development of the cilium, and those associated with this disruption of the trafficking within the cilia. And when you look at some of the common syndromes, there are in fact 3 of them that I've highlighted here: orofacial digital syndrome, Joubert syndrome, and Palliser-Hall syndrome, that actually include hypothalamic hamartoma as a feature. So that further suggested this link between the development of these tumours and potentially a defect in the cilia. So this brings me to the hypothesis and aims of this project, which is a project from Timothy Green, who was an honours student, is now a PhD student in my lab, and it's been the main focus of his thesis to date. So our hypothesis was that sporadic hypothalamic hamartoma could be due to these somatic single hits, as had already been demonstrated in sonic hedgehog genes. But in addition, there could also be these germline somatic double hits contributing in a recessive fashion within the lesion, and not in other parts of the body, was our hypothesis. And so the aims were then to identify pathogenic variants in both the known genes, but also use exome-wide approaches to explore all of the cilia genes to identify more genes with these germline and somatic double hits. So for this study, we had, obviously our previous cohort was 38 cases, and we'd solved 14 of those. And we had still had the data available from 24 of the unsolved patients, both exome data as well as chromosomal microarray data to look at copy number changes as well. And then in addition, we were able to add a further 28 new patients to the cohort, all of which had high depth exome sequencing. So we had a total cohort of 52 patients with about half of them with brain tissue available and about half of them without brain tissue available. So in terms of the results from the exome sequencing analysis, so initially we obviously looked at the known genes, and we were able to pull out 4 cases with mutations in the well-established GLE3 transcription factor of the sonic hedgehog pathway. We're also able to, and I'm sorry, it keeps moving forward for some reason, also able to identify 3 cases in another known gene, OFD1. These were all male cases. It was an excellent gene. So we had 7 cases with dominant mutations in known genes. And we then looked across all of the known and even candidate cilia genes, and we were able to identify biallelic cases. So firstly, 2 cases in the DINK2H1 gene, which confirmed the findings of the Japanese study, both with a germline and somatic change. And then in addition, we were able to implicate a new gene, WDR60, also a cilia gene, with 3 cases with biallelic variants, IFT140, and the smoothen gene, which is a gene in the sonic hedgehog pathway. And interestingly, all 4 of these genes had been associated with different ciliopathies. So in 3 of them associated with subtypes of short rib dysplasia with or without polydactyly. And the smoothen gene, as I'm going to show you a little bit later, has been now associated with a Palliser-Hall-like syndrome as well. In addition to the confirmed cases, we also had additional cases with candidate variants, either a germline or a somatic variant, where either we didn't have access to brain tissue to look for that somatic second hit, or in the case of the somatic variants, where we didn't have sufficient DNA to go to search for, the germline second hit. But we suspect these cases would have a second hit. And the issue we also had was, given this was largely an historical cohort, we also had, in some cases, minimal DNA to work with. So I just want to give you some examples of the 2-hit model. So this is the DINK2H1 gene, and one of our cases had somatic and germline single nucleotide variants. So both one a missense change and one a somatic nonsense change. And of course, one of the important things that we had to demonstrate was to show that, in fact, these variants were present in trans, obviously, within the hamartoma tissue to fit with our idea of this recessive model just within the tumor. So, and of course, trans is the configuration we're interested in. Obviously, cis, where we had both variants on the same allele, would not fit with our model. So as an example of how we're able to show that the variants were in trans, so here for WDR60, as I said, there were 3 cases. This is one of the cases where we had a germline frameshift variant and a somatic copy number loss variant. So when we did Sanger sequencing, we could clearly see the frameshift variant in the blood, showing that obviously the germline variant. But when we looked in the hamartoma tissue, you could see that the frameshift was present, but at much lower level than it was in the blood. And that's because there was obviously the somatic deletion on the other allele only within the hamartoma. Similar story for the IFT140 case. Again, in this case, a germline frameshift variant and a somatic loss of heterozygosity variant. Again, a similar pattern on Sanger sequencing showing this reduction of the frameshift variant via Sanger in the hamartoma tissue compared to the blood. So we're able to confirm that they were in trans. Now, most interestingly is when we looked at the function of these proteins is that they're all involved in retrograde transport. So this is transport of molecules from the tip of the cilium down to the base. So IFT140 and DINK2H1 and WDR60 are all part of IFT-A or dynin subunits. And so you get if you disrupt those genes, then you can disrupt retrograde transport. Of course, given that the sonic hedgehog pathway is dependent on the cilia trafficking for normal signaling, you also get disruption of the SHH signaling response in the cilia as well. And we knew this from previously published data. So there was a study by Okbuena and colleagues back in 2011 published in Nature Genetics. And what they did was to actually generate knockout mice, DINK2H1 knockout mice. And they showed that the knocking out in a homozygous fashion, DINK2H1 was actually lethal. So these mice died mid-gestation. But fortunately, they were able to actually get the embryos prior to the lethality, and they're able to study the cells within those embryos. And they're actually able to show that knocking out DINK2H1 specifically disrupted retrograde transport. And this really had two impacts. Firstly, it disrupted sonic hedgehog signaling. So you would get a buildup of these sonic hedgehog proteins in the cilia. And the second effect is you had this bulging architecture, abnormal architecture of the cilia as a consequence of the buildup of these sonic hedgehog and potentially other molecules in the cilium. So there was two defects associated with knockout of DINK2H1. The other thing that we wanted to do was to try and determine whether or not there was an increase in the burden of rare variants in our known cilia genes in our HH cases versus ethnically matched controls. And so we did attempt a burden analysis using the known cilia genes, but also candidate cilia genes as well, sonic hedgehog pathway genes. And we also included ion channel genes as a control because there's no evidence that ion channel genes should be disrupted in hypothalamic hamartoma, although we know that ion channels can be disrupted in other types of epilepsy. So for the burden analysis, we actually broke down the germline variants that we did identify. We didn't include the somatic variants simply because we just didn't feel we had enough somatic variants to achieve a significant signal. And in fact, as you'll see, we unfortunately didn't even have enough patients to get a significant signal for the germline variants as well. But we think there is a trend. So when you break them down and you separate out the protein truncating variants and specifically the ultra rare versus the rare. So for a recessive condition, we're actually most interested obviously in rare PTVs rather than ultra rare. And ultra rare variants are the ones one would typically see in dominant disorders, highly penetrant dominant disorders. But for recessive disorders, we're more interested in those rarer PTVs. And so what you can see here is not a significant signal, but we think a trend, and we did a calculation actually. We had about 52 patients, as I mentioned in the cohort, but in order to get a significant signal, we would have needed over 80 patients. But as you can see here, it doesn't cross the line, but it's pretty close, we think, to significance. So suggesting that there may be enrichment for PTVs in the known cilia genes. So just to summarize our work on recent work on sporadic HH. So as I mentioned, we confirmed variants in 7 cases with the known genes, and these are obviously dominant somatic variants. We also were able to describe 7 biallelic cases with dominant and germline and somatic variants in 4 genes, one of which DINK2H1 had been reported, but 3 new genes, including 2 cilia genes and the smoothen gene, which is a regulator upstream in the sonic hedgehog pathway. I mentioned already the burden analysis, and so our overall solve rate for the cohort was about 27%. But if you subtract those cases where we only had blood-derived DNA available, if we had tissue, we could almost solve 50% of our cases. And so really one of the challenges we face going forward, I mentioned that this was largely an historical series. The problem we have now is the standard of care is no longer resection for these lesions, it's actually laser ablation. And that of course means that we don't get access to the resected tissue anymore, but we're doing some work now, which is still preliminary, which is why I'm not presenting it today, but where we're actually getting small amounts of tissue from the laser sheath that's actually inserted into the hematoma prior to it being ablated with the laser. We're actually washing small amounts of tissue off the sheath, and we're then using kits to actually amplify up the DNA prior to exome sequencing. And we've just got some new data on a couple of cases where we've got this laser sheath tissue, and we're optimistic that we might have enough DNA from that tissue to pick up variants, pathogenic variants, without getting access to the resected tissue, and that would be a major advance for us if we can pull it off. Now, I mentioned just at the start of when I introduced hypothalamic hamartoma, that can also occur as part of a syndrome. It doesn't always occur sporadically. And in fact, there are 3 different syndromes: Pallister-Hall, orofacial digital syndrome, and Joubert syndrome, that include hypothalamic hamartoma as a feature. And there have been a couple of genes, including GLE3 for Pallister-Hall syndrome, but also C-plane1, which is in fact a cilia gene that have been implicated as a cause of these syndromes. And in addition, most recently, there's been 2 papers describing biallelic germline variants in this same SMO gene that we identified in our sporadic cohort, associated with what's described as a Pallister-Hall-like syndrome. So during our work on the sporadic cases, we were contacted by a colleague in Germany who actually had an interesting family with 2 siblings with a familial PS or Pallister-Hall syndrome-like disorder. And you can see the pedigree here, showing that both siblings had hypothalamic hamartoma. They also had post-axial polydactyly, which is obviously a feature of Pallister-Hall syndrome, and they both had microcephaly as well. Now, they had a clinical exome sequencing analysis of blood-derived DNA from all 4 individuals. Of course, here we're suspecting, because it's a syndrome, we're suspecting germline biallelic contribution. We looked at GLE3 as the known gene for Pallister-Hall syndrome, but it was negative based on that clinical data. So what we did then was reanalysis of the exome data, and specifically based on the data in literature, looked at the smoothen gene. And in fact, we were able to pull out, firstly, a missense variant that's predicted to be damaging, and as you can see is very rare in nomad, and it's not seen as a homozygote. And then when we looked harder, we were also able to pull out, and in fact, in a synonymous change, a very rare synonymous change, again, not seen as a homozygote. And in this case, the variant is predicted to disrupt splicing. So when you put it through Splice AI or other in silico tools, the prediction is that it potentially leads to exon skipping, and it's located within exon 8. Now, we haven't been able to confirm the splicing defect yet, because obviously both the siblings are in Germany, but we're just this week post-COVID or post-COVID lockdown over in Germany, we're going to be able to get a skin biopsy from the proband, one of the probands, and also from mum, who has obviously transmitted the splicing variant. We're going to generate fibroblasts where we know smoothen is expressed, and then we're going to hopefully be able to confirm that splice defect. And we're pretty confident that we'll be able to confirm it, because when we've looked at the phenotypes of these 2 siblings compared to the reported cases, we think there's pretty good concordance. So here's a list of the major features of the 2 siblings. You can see both have developmental delay, Gelastic Seizures, obviously HH, as well as microcephaly, and then polydactyly amongst other features. And when you compare those features to the reported cases, which are listed on the table here, you can see that there's a fair degree of overlap, not with every patient reported in the series, but with many of the patients, you can see that there's shared features. So we're pretty confident that when we do get the fibroblasts from one of the siblings and from the mum, that we'll be able to confirm that there's a second hit splicing variant, and that the smoothen gene is probably responsible for the Pallister-Hall-like syndrome in these siblings. So just to conclude from this part of the talk, which really focuses on what we can do when we've got, for the most part, on what we can do when we've got tissue available. So we've shown that there's a complex genetic architecture for the sporadic form of the disease, both dominant genes with single somatic hits, but also these somatic and germline biallelic variants, and more in a recessive mode. And I should point out that this is not the only brain malformation associated with epilepsy where we've now seen these biallelic, somatic, and germline variants. There are others, such as for focal cortical dysplasia. There have been genes with these dual contributions identified as well. obviously talked about the syndromic form, where we can also identify germline variants in the smoothened gene, and overall, really, for both the sporadic and the syndromic forms of the disease, we think that the genes involved are either the sonic hedgehog signaling molecules themselves, or that there are defects in trafficking genes within the cilia that also have an impact on sonic hedgehog signaling. So I now want to switch gears for the last part of my talk and to talk about strategies that we can use in the absence of brain tissue. So far, I've talked to you about what we can do when we get access to resected tissue, but the reality is, even for epilepsy, only a minority of patients, less than 5%, actually are candidates for surgery. And so for most patients, we don't have tissue available. In addition, as I've mentioned, there is an increase in terms of, for both efficacy and safety reasons, an increase in the number of ablative approaches rather than resective that are being used, and HH is a good example. So what we really need are alternative, accessible biological sources for diagnosis. And we've been working on a number of these approaches as part of an NHMRC grant, and more recently an MRFF grant. And today I'm going to focus on the one we've got most data on and that we've recently published, and that's our cell-free DNA cerebrospinal fluid liquid biopsy. And again, even as with the tissue, it's an approach that relies on very sensitive techniques. So we need high depth sequencing, at least 200-fold, but preferably higher. And of course, we need sensitive methods for validation, such as droplet digital PCR, which I'll talk briefly about. So really the idea for this work, as with many ideas that we have in genetics, came from work in the oncology field. So back in 2015, this US group published a paper describing detection of tumor-derived or tumor mutations using tumor-derived DNA from cerebrospinal fluid. So in their series, I think they had somewhere around 30 patients. They were able to identify both driver and secondary mutations in these derived from these tumors in cerebrospinal fluid cell-free DNA. So we know, it has been known for some time now, that tumors can shed DNA into the CSF, and this group showed that they could pick up about 90% of the patients. They were able to pick up the mutation in the CSF, and the 10% that they weren't able to pick up was because the tumors weren't adjacent to a CSF reservoir. So we thought that we might be able to apply the same approach, but instead of a malignant disorder, that we would apply it to a non-malignant neurological disease for the first time. And one of the reasons we thought about this is because of our work on hypothalamic hamartoma. We knew that the hypothalamus forms the base of the third ventricle, which is one of the major CSF reservoirs in the brain. So we thought if there was a variant in these benign tumors of the hypothalamus, that variant might be able to make its way into the CSF reservoir nearby, based on the fact that there is cell death going on in these tumors, although of course not to the level that you would see in a malignant tumor. And I should mention that there's obviously two ways that we can access the CSF. One is in a more invasive way through neurosurgery. You can collect it with a dural puncture, or less invasively, we can do it without requiring surgery in a procedure known as a lumbar puncture. So that really sort of sparked our interest in trying to develop a CSF liquid biopsy for epilepsy. And this is work by another PhD student in my lab, Daisy Yi. And so the background really is that we know that clinical liquid biopsies are routine in certain specialties. So we know, for example, it's used in obstetrics routinely. We've heard of the Harmony tests. So now when we're looking for prenatal variants, it can be done using the cell-free DNA derived from the fetus, but it's obviously located within the maternal serum. For oncology, we know that there are liquid biopsies that are used to both detect but also monitor mutations that cause cancer. And of course, for transplantation, these liquid biopsies are used to compare host with donor-derived DNA to determine whether or not there is rejection of the transplant. So the CSF liquid biopsy relies on this biological phenomenon whereby, following cell death, such as apoptosis, short fragments called cell-free DNA from the cell actually enter into the bodily fluids. And these are all around 170 base pair in length. They're obviously, in the case of the CSF, what we suspected was that they were externalized and shed from brain cells into the CSF. And we actually went on to show that, and I'll talk about that data. And obviously such a biopsy was already established, as I mentioned, for brain tumors, but we wanted to apply it to a neurological disease, obviously a non-malignant one. So the research questions we had were, was there sufficient DNA in patients with epilepsy in their CSF to make this viable? Could we show proof of principle? We were also interested in whether or not the levels differed between epilepsy patients and patient and control patients that we were able to get CSF from. We're also, of course, most interested in whether or not we could detect somatic mutations through the CSF cell-free DNA. And then finally, we asked, is this DNA derived from brain cells? So to answer this question, we were able to put together really two cohorts. And these cohorts are really based on the restrictions of our research ethics. So we were able to obtain CSF from a cohort of patients with epilepsy. These were all children who were undergoing neurosurgery at the Royal Children's Hospital, and the CSF was collected by dural puncture prior to the resective surgery. We also had a cohort of controls that were collected at the Austin Hospital. These were adults who had lumbar puncture, and they were having lumbar puncture for a reason unrelated to epilepsy. They didn't have epilepsy. They, for example, had migraine and they needed a CSF to check to see if there was any central nervous system infection. So we're able to collect 28 of those samples. We then extracted the cell-free DNA using standard QIAGEN kit and then performed Digital Pcr to quantitate the amount of cell-free DNA, which we expected would be at very low concentration. And we used an established assay in the lab, just looking at the wild-type reference allele for a particular variant in the gene. So I mentioned droplet digital PCR before. So we use this because it's an incredibly sensitive method. For those of you not familiar, it's actually about 10-fold more sensitive than ultra-deep sequencing, down to about a possibility of 0.01% allele frequency. And it's a method that, as you can see, relies on very specific, the design of very specific probes that are labeled with different fluorophores. And typically you would have a probe targeted at a mutant sequence and compare that to a probe targeted at the wild type or reference allele. And the advantage of this approach is that it uses, the first step in the process really is to use a vacuum to actually push the DNA combined with the PCR reaction into individual oil droplets. And the idea is to end up mostly with individual single DNA molecules in each of those oil droplets. So it provides very high resolution. We then do a bulk PCR to amplify up the DNA, and then put the samples through a droplet reader to measure the fluorescent signals. So it has a number of advantages. It can obviously target specific mutations if they're known. It can also be used to determine copy number, where you don't necessarily have to know the specific copy number change, but you can target specific regions of a gene to see whether or not there is a deletion or maybe an insertion. It's really good for poor quality DNA. So even old FFPE samples that are degraded, we can often have success detecting variants with that type of tissue. And it's relatively fast and inexpensive, particularly if you're looking at recurrent variants, which can be the cause of lesional epilepsy. It has some limitations though. It obviously relies on short amplimers. There's very limited multiplexing because there's only a few fluorophores available for these probes. And of course it relies on prior knowledge. So it's not really a screening technique. You have to know what you're looking for. So as I said, we used this approach to quantify the low levels of cell-free DNA that we suspected would be present in the CSF. And this is just one example comparing a patient to a control. And this is the typical sort of readout. Here we're only, of course, looking at the reference allele, which is, or the wild type allele, which is always shown in green. If we're also looking at a mutant allele, you would see blue droplets, and you'll see what those look like in a moment. So this was the summary of the results. When we looked across our cohort of 28 patients and 28 controls, we did actually see an increase in the amount of cell-free DNA in our epilepsy patients versus controls. However, one has to recognize that this is still a relatively small number, and we are collecting more patients. And we also have to acknowledge that some limitations here, where we've got obviously a different approach to collect the CSF and the different age of the patients versus the control. So there are definitely some limitations, and we need to do further work to really verify whether or not this difference we're seeing is real. And the reason that that's so interesting is that one hypothesis could be that the seizures in the patients actually lead to an increase in cell death, and that might explain this elevated level of cell-free DNA. But as I say, at the moment, that's only a theory, and we need to do more work. In terms of the molecular diagnosis, so we actually had 3 patients where we were able to demonstrate proof of principle to detect the variant in the cell-free DNA from the CSF. So we had one patient with focal epilepsy and a condition known as bilateral double cortex and a mutation in the LIS1 gene, which is a known gene for that particular condition. The second patient, also with focal epilepsy, had focal cortical dysplasia and a mutation in the TSC1 gene, which is again a known gene for that condition. And in the final patient, patient three did not actually have a variant at the time that we tested the CSF, but because we had prior knowledge that around 60% of patients with ganglioglioma actually have a recurrent variant, this V600E variant in the BRAF gene, we're actually able to screen that gene with a GDPCR assay, and we were fortunate enough to pick it up in this particular patient. So the strategy was that we had paired brain, blood, and CSF cell-free DNA samples from all 3 of these patients. And then we did individual designed or used a commercial GDPCR assay for all 3 of these mutations. And we were able to determine the detection limit for all of them. We do this using G-blocks, which we get from IDT. And we're able to show that these assays were able to pick up alleles down to 0.025% allele frequency reliably. So I just want to go through each patient quickly here. So this is the first patient with a LIS1 variant that we previously in both blood, saliva, and indeed in brain in previous publications. And you can see that we were able to pick it up at about 9.4% frequency in the CSF cell-free DNA. Here you can see in the 2D plot for the ddPCR results showing you the positive blue droplets, each one of these droplets obviously representing an individual DNA molecule, and then you compare that to the wild type droplets in green, and obviously the percentage is based on the ratio between those two. The second patient had a variant in TSC1 that was initially detected in brain-derived DNA at around 2% frequency. It was absent in blood, so this is we think a brain-specific somatic variant, and we were able to pick it up at about 7.8% frequency in the CSF cell-free DNA. And here we think the ratio is actually seen higher in the CSF just because we had such a low amount of DNA from this particular CSF sample. And then the final patient that I already mentioned with ganglioglioma, so in this case had no variant at the time of testing of the CSF, but given our prior knowledge about the BRAF V600E contribution to the ganglioglioma, we decided to test for that variant, and we detected it at 3.2% frequency in the CSF, and subsequently validated it in brain-derived DNA from this patient at about 20% frequency. And again, this was absent from blood, so another brain-only somatic variant. So having confirmed that we could pick up these somatic variants in the CSF, we then were also interested to confirm whether or not the CSF cell-free DNA that we'd isolated was in fact brain-derived. So we collaborated with Ryan Lister's group at University of Western Australia and also Zach Chatton's group at University of Sydney to use a method that they had developed called methylation haplotyping, which relies on differential methylation markers that are present in some tissues but not others. And what they did was to take 5 of our CSF samples and also 5 blood or plasma-derived cfDNA samples that they had access to, and to perform whole genome bisulfide sequencing to essentially pick up these methylation markers. They looked at markers across different tissues, but they then really focused in on markers of neuronal tissues such as NUN, and also markers of plasma as well. And what you can see from the graphs here is that when they looked at the CSF samples, they showed that it was enriched for CNS-derived markers, whereas when they compared to the blood or plasma-derived cell-free DNA, they saw enrichment for plasma markers and not for the CSF markers. So we think, based on this data, that the CF DNA is mostly brain-derived, which is obviously important given that we've already picked up these mosaic variants, and we wanted to be sure that they were most likely present in brain-derived material. So I already mentioned some of the limitations with this work in terms of the route of collection and also the age of participants. We have a number of future directions that are ongoing. So we're very interested now in, obviously exome-wide analysis. Given so far we've only done targeted analysis, we'd like to be able to pick up new mutations in these patients. And we're both interested in lesional patients that I've talked about who have, an obvious lesion on imaging and pathology. But we're also interested in the much more common focal epilepsy patients who are actually non-lesional, have no findings on imaging. And we're interested for the first time to really be able to look at the somatic contribution to those patients, because being non-lesional, they're never going to be candidates for surgery. We don't normally get access to the tissue. And what we're planning to do with our research ethics here at the Austin, which enables us to now collect CSF samples on a research basis from patients with epilepsy. So we're going to do lumbar punctures on our non-lesional patients and interrogate that CSF for somatic variants. We're also quite interested in this theory around whether or not seizures contribute to an increase in cell-free DNA in the CSF. Now, one of the challenges we face is we can't do lumbar punctures on patients obviously who are having seizures, because it's quite a dangerous thing to do. So what we're going to do instead is collect blood samples pre and post seizure when patients come into the ward here at the Austin for monitoring. So we'll have baseline samples prior to the seizure, and then we'll have samples collected post seizure, and we're going to compare the amount of cell-free DNA to see if there is an increased post seizure. And I should also point out we have some preliminary data. This is actually data generated at AGRF, showing that we can do exome-wide sequencing on these very low-level CSF samples. The challenge, of course, is, as you can see, the number of duplicates that we obtain, because we have to do extra rounds of amplification. So we're now working on trying to optimize our exome approach to try and reduce the number of duplicates and therefore increase the coverage of the exome sequencing. But I mean, the good news is when you look at the figure there, you can see that the insert size matches with the fragment size that we expect for the cell-free DNA in the CSF. So we're confident that with further optimization, that we'll be able to get high-depth sequencing from the cell-free DNA in the CSF as well. And that's really a subject of a number of grant applications, including one that just went in recently. So I'll just finish by highlighting some of the impacts that this can have on clinical care and practice. So we know that by detecting these somatic variants that we can confirm the molecular lesion. And this, of course, informs the clinical diagnosis and etiology. It can end the diagnostic odyssey for these patients. It can also provide insight into the molecular pathology, and we can go back also to the imaging findings and try and correlate it with the radiological findings. For example, the size of the lesion, looking to determine whether or not the level of mosaicism correlates with the size of the lesion. It also provides specific target information for intervention. For example, there are inhibitors to mTOR that are already clinically approved, such as sirolimus. And it might be for that subset of patients that don't benefit from surgery, it might be that they could have, in addition to the surgery, they could also have therapy with these inhibitors to try and reduce the size or the effects of these lesions. So I'll just finish by acknowledging all the different groups involved in the work, and of course, as well, AGRF for their support in terms of the sequencing strategies that we've used for many of our studies. So thank you for your attention, and I'll happily take any questions.
Okay, excellent. Thanks. Michael, thank you so much. I was a neuropathologist until about a year ago, so I found that fascinating.
Thank you.
I just wondered, have you or has anyone looked at polymicrogyria in the context of the study?
So that's a good question. So we do work on some cases with polymicrogyria, although Rick Leventer's team at the Children's Hospital works on many more, and has in fact just published a new paper that's just come out in Brain Communications. It's online at the moment, on a large series of PMG cases that he's analyzed, but I think my understanding that's mostly blood-derived DNA he's looked at. So the issue we face with polymicrogyria is that most of the patients don't actually have resective surgery, so we don't normally get access to the brain tissue. But Rick has just reported a number of genes that he's identified in blood. As you may be aware, unlike say focal cortical dysplasia or some of the other lesion types, the genes haven't fallen out as easily for PMG, but you're right in suggesting it as a potential target for this approach, because obviously, we can look for those genes that Rick's now found in the CSF, and maybe that might increase the yield.
Sure. Okay. And what about the tissue? The microdissection and so on. Because I know of a few cases that really were basically not worked up, or at best very poorly worked up.
So yeah, so for some of the cases, particularly the childhood cases, they do have multiple resections, and sometimes, Rick has done some nice work where he's been able to dissect out different, particularly for focal cortical dysplasia, dissect out different regions where the lesion has infiltrated, and then look at the gradient, the mosaic variant gradient in those different regions, and in fact has shown differences. One of the other nice things he's done with FCD is also you can laser dissect out the dysmorphic neurons, and then you can look at the frequency just within the dysmorphic neurons. And what he's actually showed in a recent paper, which I think was in Annals of Clinical Translational Neuroscience, he showed that the variant is only present in the dysmorphic neurons in at least one FCD patient, but not in normal neurons that he was also able to laser capture, dissect.
Yeah. And I think that approach, the abnormal versus normal cells, whatever, if they glia or neurons, usable in a lot of these things like gangliomas and so on.
Yes.
Which got mentioned, and
yep.
Again, I think relatively understudied as an opportunity, and quite a no-brainer. Yeah? And
the big question we really want to answer is, do we see the variant, or do we see it at lower frequency in healthy tissue than we do in the pathologic tissue? We've wanted to actually do it for some time with the hypothalamic hamartoma cases, but we never get normal tissue in the children, although there are rare cases of fetal hypothalamic hamartoma, and we've recently obtained a couple of those cases, which we're hopeful will be able to, as you say, dissect out the abnormal hypothalamic region, but also, for example, in one of the cases we're looking at now, that we hope to be able to get pituitary, dissect that out from the block, and then compare, and that might answer that question. We are also aware at least one collaborator that has a post-mortem brain sample as well from a HH patient, so we'd love to analyze that as well to try to answer that really important question, I think.
For sure. And just have you given much thought? This is obviously not your main area, but just I was always intrigued about how these seizures actually started and then propagated in certain of these strange lesions that we encountered. And hypothalamic hamartoma is no different. Do you have much of an idea of how it might propagate?
Yeah, so I mean, yeah, you're right. It's not my area, but we have had a few conferences where we've heard pathologists present on with theories around how that might happen. But as I understand it, at least for hypothalamic hamartoma, we've got no sort of concrete data to show how those seizures propagate. How you go from a very specific focal seizure in one very small part of the brain into this generalized phenomenon. So the answer is we don't have the data, but I know that there are people looking into it.
Okay.
I've got a question, which was, I guess, partly covered about, in your discussions, the number of cells with variants, etc. Is there any correlation between, I guess, the load of somatic variants and the severity of disease or onset of disease? Is there any work done in that?
Yeah, so there is some evidence, particularly around lesion size. So there is some data suggesting an association with the higher mosaic variant frequency and larger lesion. That's particularly data from tuberous sclerosis, another lesion type with these tubers develop in the brain, but they can develop in other areas as well. And there is evidence from looking at those studies that there is a correlation between the size and the mosaic variant frequency. But there's still more work to be done on the other lesion types as well. And then in terms of severity, that can be more difficult to interpret. I'm not sure that there's strong correlations with variant type and frequency, but it's something we're planning to look at as part of the larger NIH grant we've put in to actually try and answer some of those questions as well.
Okay, so if we don't have any other questions, we might wrap that up. And I'd like to finalise by saying thank you very much, Michael. It was a fantastic presentation, and we really appreciate the time that you've made available to everyone here. So thank you very much.
Thanks, Matt. Thanks, everybody.
Thank you very
Okay.
much.
Thanks, Michael.
Bye.